Hello, my dear student, with a new lesson of biology. Are you ready? Let's start. Today, our lesson, this is about wonderful things. This is about structure and function of antibodies. Lesson objectives. You must recognize the concept of immunity and its importance. Describe structure of an antibodies and its mechanisms. So, look at to this picture and imagine. Did you know many dangerous microorganisms in our environments? Did you have any idea about how our body can defenses the microorganisms? Today we're gonna to talk about all of these. Let's see. Human is exposed to continuous threat like pathogens, including some insects, animal protozoa, fungi, bacteria. So, God gave the human immune system to defend these dangers. There are two systems of immunity in man. This is number one, innate immune system, and number two, acquired immune system. The innate immune system, this is the first line of that defense, like skin, mucous membranes, saliva, flushing action if you're in tears, and stomach acid. This is stops infection because it enters to the body. If these infections are pathogen entire to the body have that second immune system. This is called acquired immune system. This is composed from B cell immunity. This is called humoral immunity, and T cell immunity, cell mediated immunity. B cells, that's many responses. But today we're gonna to talk about the antibodies. Antibodies. Definition of antibodies. Antibodies are specific protein known as immunoglobulin. They are five types: IgM, IgA, IgG, IgE, and IgD, which circulate in blood and lymph. This is a type of immunoglobulins or antibody, and we can collect this is in the word MAGID. So here, IgG antibody, this is a 1 unit, IgA 1 unit, IgE and IgG, this is only 1 unit, and the IgM, this is the only pentamer, this is composed from 5 units. So it can attachment by more than one antigen in one time. Here, surface of bacterial cell that invade body tissue have compounds called antigens. Receptor on the surface of B lymphocytes recognize and join with antigens on their surface of bacterial cell or foreign bodies and produce antibodies. B lymphocytes can invade the antigen on the surface of microorganisms and the other foreign molecules by producing antibodies which circulate with the blood and lymph. Even structure of antibodies. Antibodies are proteins called immunoglobulins that are Y-shaped and present in the blood and lymph of human and other vertebrates. They are produced by antibody secreting plasma B cells. The antibody consists of two pairs of polypeptide chain. Two of these chains are long and called heavy chains. The other two chains are short and called light chains. The four chains are joined together by this disulfide bonds. So here, each antibody has two identical antigen binding sites. The shape of these sites is different from an antibody to another. The binding between the antigen and its specific antibody and this site resemble to lock and key. The binding from an antigen form an antigen antibody complex. The antigen binding sites are located at the two types of Y-shaped molecules and is known as variable region V. Because the shape varies from antibody to another, while the rest of the antibody known as the consonant region because it has consonant shape and structure in all types of antibody. The specificity of antibody is determined by the conformation of amino acid and the antigen binding site, the variable region of the polypeptide chain, which is a mirror image of a specific antigen. Antigen. So here, this is a definition of antigen. Surface of pathogen bacteria cells and viruses that invade body tissue have compounds called antigen. Mechanism of antibodies. We have five mechanisms of the antibodies. Number one, neutralization. Two, agglutination or clumping. Three, precipitation. Four, lysis. Five, antitoxins. The first one of these mechanisms, this is called neutralization. The antibodies bind to the other outer coat of viruses that prevent the viruses from the adhering to the membrane of host cells and from spreading or passing into them. If the viruses succeed in penetrating the host cells membrane, the antibody will prevent the nucleic acid of the virus from coming out of the protein coat and replication inside the host cells. Agglutination. Agglutination, this is meaning form a clumping. So some antibody like IgM antibodies, this is a pentamer in its structure, which bind to more than one microbe or antigen. This leads to the clumping of microbes on the same antibody. This makes them weaker and liable to engulfed by phagocytes. Precipitation, this is a third way of a mechanisms of antibody. So the difference between precipitation and the clumping or agglutination, this is only here, this is a soluble antigen. So number three, precipitation, happens usually in the soluble antigen, in which the binding between the antibodies and this antigen lead to formation of insoluble antigen antibody complexes, which form precipitate to facilitate engulfing by phagocytes. For lysis, the binding between antibodies and the antigen activates specific proteins and enzyme called complements to lysis the coat of antigen and dissolve their content, which makes they easily engulfed by phagocytes. The last one, this is antitoxins. So antibodies can also bind to toxins and form complexes. These complexes activate the complement to react with them, which lead finally to detoxifying them and also makes them readily engulfed by phagocytosis. Here we have some question. We're going to answer about it. The first one, which of the following label highlights structure that can differ between RNA and DNA? A, 2 and 3. B, 3 only. C, 1 and 2, B, 2 only. Let's review the structure of DNA and RNA in the first. DNA and RNA are polymers composed of the numerous subunits known as nucleotides. DNA and RNA nucleotides are composed of beta sugar. Here, this is number 2, phosphate group, this is number 1, and nitrogen base number 3. However, in DNA, the pentose sugar is deoxyribose, but in RNA, it's ribose. Adenine, thymine, cytosine, and guanine are the nitrogen base found in DNA, but uracil replaces thymine in RNA. DNA forms a double-stranded molecule, while RNA forms single-stranded molecule. So there are some differ. This is in DNA and RNA. A deoxyribose sugar is in the pentose sugar found in DNA nucleotide, where the ribose sugar is found in RNA molecules. So we can choose the correct answer, which D, 2 only. The differ, it's only in structure D, the pentose sugar. In DNA, this is a deoxyribose, and RNA, this is a ribose. The next question, which of the following structure does the oxygenated blood pass through? A, structure ZNW. B, structures Y and X. C, structure Z and X. D, structure W and Y. If we remember the circulation of blood in the heart, we see the pulmonary vein coming from lung pushes the oxygenated blood into the left atrium. The atria contract and the right atrium pours the deoxygenated blood into right ventricle, and left atrium pours the oxygenated blood into left ventricle. Then the ventricles contract, form right ventricles. The oxygenated blood follows through the pulmonary artery to the lungs. And from the left ventricle, the oxygenated blood is distributed through the largest artery, the aorta, to all parts of body. Now, the correct answer, number A, right, structure Z and W. The next question, in a hypothetical situation, if all Bowman's capsule and both kidneys of a person stopped working. A, the urine will contain blood. B, there will be no urine. C, the urine when contains large water quantities. D, the urine will be full of urea. If you're gonna to review the formation or structure of the Bowman capsule, we remember that the first processing of formation of urine is a filtration by the Bowman capsule. So if Bowman capsule damages, there will be no urine. So the correct answer, this is B. There will be no urine. The next one, the Grad fellow measured the concentration of glucose, insulin, and glucagon respectively in a man over a 42 days period. This is a chart of concentration of glucose, here concentration of insulin, and this is concentration of glucagon. Which of the following illustration description the man? This man has damaged alpha cells. This man is on long fast. This man has damaged beta cell. This man is diabetic. Let's review in the first control of blood glucose by hormones, insulin, and glucagon as by negative feedback. Glucose is a form of simple sugar. Glucose is classified as high energy molecule due to its several chemical bonds during cellular respiration. These bonds are broken, and the energy released is used to power the life function of cells. This procedure is what makes glucose vital for cells to operate properly. The glucose concentration blood must be in normal range so that the level of glucose at its normal level controls the hormone insulin and glucagon. Insulin stimulates the liver to absorb glucose, which is then stored as a substance called the glycogen. Glycogen is an insoluble complex carbohydrate, contains many glucose units. Glucose can be converted into glycogen and stored for future use. Glycogen is also stored in muscle cells. Insulin also there our bodies to turn glucose into fat, which then deposit in fat cells. Also, insulin promotes cellular function that use up glucose, such as cellular respiration, as insulin causes body to use and store glucose to concentration of glucose declines. So here, when blood glucose levels drop, the pancreas shut down insulin production. When cells in pancreas detect difference, they produce hormone known glucagon. Glucagon causes cells in the liver to turn the glycogen back into glucose and release into the bloodstream. Also, we know when blood glucose level drop, the pancreas sense this and raise amount of glycogen in bloodstream. That glycogen causes our cells to store and release glucose, increasing its concentration in the bloodstream. The pancreas detects the increased glucose and eventually lower the concentration of glucagon in the bloodstream as a condition back to regular. So the correct answer B, the man is on long fast. The next question, the diagram shows the presence of fibroid benign tumor in female genitalial system. Which sentences could be correct regarding this woman? A. She has no ovulation. B. She will be completely infertile. C. She has difficulty in zygote implantation. D. She has excessive ovulation. So, first we will review the diagram. The female reproductive system organs are two ovaries, contain immature egg cells, and secrete many hormones like estrogen and progesterone that control the menstrual cycle. The second organ, this is the fallopian tubes. This is two tubes are connect each other by uterus. Now, how do pregnancy occur? Egg cells mature and are expelled from the ovary and fallopian tube. Fertilization occurs in the fallopian tube if a sperm can pass through the female reproductive system and paint with the mature egg cell. If fertilization happens, the zygote will implant here in uterus. So a female has a fibroid in this uterus. She has difficulty in zygote implantation. Now, the correct answer, yes, number C. She has difficulty in zygote implantation. Now, this is the end of our lesson. Thank you.